So good evening, everyone. If you are new to Illinois RiverWatch Network, we are a citizen science organization that trains volunteers to monitor water quality in their own communities. The mission of RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. Before we get started, I ask that you keep your microphone muted during the presentation to limit the distractions. We will have a Q&A session after the presentation, so if you have a question along the way, you can either put it into the chat box to be answered at the end, or you can save the question and ask it aloud. And with that, I would like to welcome Megan Stropis. Megan is currently in her last semester at Joliet Junior College. She will be graduating this spring and plans to transfer to a four-year college where she will continue to pursue a degree in biology with a biomedical science focus. Today, Megan will be talking about research that she conducted in the summer of 2021 with her two classmates, Gwen Bach and Ben Murdoch, with the advisement of Professor John Griffiths. We are pleased to welcome Megan Stropis for her presentation tonight.
Hey, thank you guys so much for having me. Let me go ahead and get my screen shared for you guys. We can get started. Okay, is everyone able to see this?
Yep, you're good.
Okay, perfect. So tonight, like Hannah said, we'll be talking about the comparison of benthic macroinvertebrate colonization of microhabitats, distance decay, and heterodendes in relation to stream quality and determination. Distance decay and heterodendes were completed by my fellow students conducting this research, but since they couldn't be here this evening, I'll be sharing some of their work on their behalf. So to begin, here's just a brief overview of what we'll be discussing. First, I'll share some general background information to get everyone up to speed and on the same page, along with going over the methodology used during most of this research. Then I'll be diving into my own research topic of microhabitat comparison, followed by Ben Murdoch's work on distance decay, and finally Gwen Bach's work on heterodendes. So without further ado, let's get into some background info. So we're going to begin by defining just what exactly a benthic macroinvertebrate is. Benthic just means bottom dwelling. Macro is something that can be seen with the naked eye. And invertebrate is just an animal without a backbone. So when we put that all together, we get that benthic macroinvertebrates are bottom dwelling organisms, which lack a backbone and are discernible to the naked eye. Their collection plays a very important role in determining stream quality. And some factors that affect stream quality are going to be pollution, soil erosion, organic wastes, nutrient enrichment, along with habitat. So within the taxa that we'll collect, the insect orders Ephemoptera, which is mayflies, Plecoptera, stoneflies, and Trichoptera, caddisflies, also collectively known as EPT, are more significantly affected by environmental changes, and their presence is more profound in better quality streams. Certain macroinvertebrates are able to tolerate pollution more than others, separating them into pollution intolerant and pollution tolerant categories, also referred to as PIT or PT respectively. They are assigned a tolerance index number which aids to calculate stream quality. So here are some images of what our EPT macroinvertebrates look like to help give you a better idea of what we're working with. Here on the left is a mayfly that we would see when we're out. In the center we have a stonefly, and on the right is a caddisfly larva. Actually, that's typically what we would find them as. So to give you some more background on microhabitats, different microenvironments exist within streams and provide varying habitats for the organisms that inhabit it. The two main ones that we sampled and are going to be focusing on this evening are riffles and snags. Riffles are areas of choppy water that form over and around stones or gravel. The rocks provide small openings for macroinvertebrates to colonize in, creating very ideal locations for sampling. Snags are fallen trees, logs, or tree roots that are submerged underneath the water. They provide an ideal location for habitat colonization due to their rigid surface for invertebrates to cling to and colonize. So now that we've had some background information, let's go into the methodology. This study was conducted at 3 locations in Will County, Illinois at Lily Cache Creek, Van Horn Woods East, Avery Preserve Disc Golf Course, and a site immediately adjacent to the established Avery Preserve location. Both Van Horn and Avery Preserve sites have been part of Illinois RiverWatch since 1998. The adjacent site was newly established during this 2021 study and immediately upstream of the Avery Preserve location. The Van Horn Woods location was sampled May 26, 2021, and was located downstream and characterized with 50% canopy coverage. The Avery Preserve was sampled on June 2nd and was located upstream and had an open canopy coverage, and the adjacent site was sampled June 9th and also had an open canopy and was upstream. These are the sites that we were staying at and worked with. So on the left, we have Van Horn Woods East, and you can see in the shade on the water just how that canopy cover looked like. And then in the center, we have Avery Preserve, and on the left, the immediate adjacent site, which those last two both have open canopies, as you can see there. So some of the methodology of what we did in the field is our biological surveys were conducted following the Illinois RiverWatch Stream Monitoring Manual. Upon arrival, we took a lot of observations such as present weather, worst weather in the last 2 days, air and water temperature, water appearance and odor, and turbidity were all determined. And then we took 4 50-foot sections, so 200 feet total, measured out, and within each the canopy cover, algal growth, substrate siltration coverage, and bottom substrate were estimated and averaged and totaled for each site. While we were out there, 2 riffles were identified and sampled, starting at the downstream-most riffle. And 2 roles were given, the netter and the kicker. A 0.5 millimeter D-frame dip net was inserted vertically immediately downstream of the riffle, with the large opening facing upstream towards the netter. The large rocks in front of the net were rubbed off and placed in a collection bucket by the kicker. The kicker then stood there and shuffled their feet in a roughly 3-foot by 3-foot area of the riffle for 3 minutes, disturbing the substrate below into the net. The net was then inverted over the bucket and rinsed with wash bottles filled with stream water to collect all the organisms that were remaining and clinging on the net. And this process was repeated at the upstream riffle, and both samples were collected into the same bucket. When we were looking for our snags, we identified those as well as either just previously those fallen logs or tree stumps. Then I would replace that same D-frame net immediately downstream of the snag while the kicker scraped the surface of the log with their hands for roughly 10 to 15 minutes into the net. That same net inversion technique and rinsing process was also repeated into a separate bucket than the riffle was kept. Both of our riffle and snag samples were subsampled due to the volume of organisms we collected. All macroinvertebrates for each sample were transferred into a subsampling pan with a 4x3 grid, similar to what's shown on the right here. The pans were filled with approximately ½ an inch of soda water. The carbonation slows the organisms down, and so they don't move as fast, making subsampling much easier. A random number generator was then used to select a square and remove all organisms into a jar of 91% isopropyl alcohol until we had at least 100 organisms collected, with a minimum of 50 from riffles and 50 from snags. Once collected, the pan was reviewed and representative organisms were selected and added to the jar if there were not any chosen through random sampling. Once we moved into the lab, identification of the samples began using a dissection microscope and different keys. The identification was checked over by fellow students and recorded according to the Illinois RiverWatch network. The total taxa, EPT taxa, and total tolerance value were determined, denoted as sigma N, sigma taxa, and sigma TV respectively. Pollution intolerant macroinvertebrates have a tolerance index of less than 5.5, whereas pollution tolerant have greater than 5.5. The macroinvertebrate biotic index, also known as MBI, was calculated by dividing the sum of the total tolerance value by the sum of the total taxa. And the Shannon diversity index was calculated by taking the summation of total taxa encountered and subtracting the quantity of the fraction of the entire population P made up of species I multiplied by the natural log of P sub I. Basically a lot of plug and chug in that one right there. The higher the index calculated, the more diverse the organisms found in a habitat are. And here we could see the macroinvertebrate biotic index that's used to determine the stream quality according to RiverWatch protocol, as shown in the table here. It is also used to find the quality of taxa richness and EPT taxa richness. So all 3 of the different research topics done, we will have different ratings based on these richnesses and MBI, and it's all referred to from this chart here from RiverWatch. So now that we've gone over some general methodology that everyone shared, we're going to go into my specific research, which was microhabitat comparison. So a little bit of more background on microhabitats. The type of microenvironment has been shown to affect the number and diversity of taxa found there. Some evidence shows that there is a greater increase in diversity in snags than riffles, some cases even finding nearly twice as much unique taxa, especially when riffle quality is poor. Snags might act as a sanctuary of some sorts to the organisms in that area. On the contrary, other studies propose that riffles have more diverse microhabitat for macroinvertebrate sampling. Multiple habitat sampling protocols are required sampling within a proportional space, which can include riffles and snags, along with submerged and baked plants and sediments. And RiverWatch uses a modified multiple habitat approach that samples from 2 microhabitats based on a hierarchy of diversity. Riffles are the most diverse, followed by leaf packs, snags, and undercut banks. So the purpose of this experiment was to compare benthic macroinvertebrate samples between varying microhabitats. Samples were taken from riffles and snags according to RiverWatch procedure at 3 locations. They are compared to each other in order to determine the taxa richness, EPT taxa richness, and water quality. The hypothesis was that snags would have an increased number of pollution intolerant and EPT taxa than riffles, therefore indicating better water quality. So this figure here expresses the total recorded macroinvertebrate taxa from riffles and snags at Van Horn, Avery Preserve, and the adjacent site. It's a little bit of a busy graph, but it does, I think, a very good visual of showing the distribution of organisms between the 6 total samples. You can see that midges, scuds, and black flies, which you can see right here, we have our scud, and then midge and black fly are right down here. So midges and black flies are pollution tolerant, whereas scuds are pollution intolerant. And then also when we look at the organisms here on the y-axis, we can see our EPT category right over here, made up of our mayflies, stonefly, and caddisflies. So that's where our EPT lie on this chart here. So this graph here compares the total number of individual taxa collected between riffles and snags, specifically at Van Horn. It was very interesting because this location had the highest number of unique taxa, which was 17 riffles and 17 for snag, but we found that it didn't share any relation to the overall number of macroinvertebrates that were collected. And then here we see again the total number of individual taxa collected between riffles and snags at Avery Preserve. And since in that first graph we looked at that scuds, midges, and black flies were in such high quantities, you'll see that that also does reflect at each individual site. There wasn't any one site that had such an outstanding number that it bumped the average up higher. And then again, here's this information of the comparison of riffle to snag taxa at our adjacent site. And it's very interesting, again, when you look through and compare them all. In this case, we had very little EPT in this location, but if we move back to Avery Preserve, there's a fair number of EPT there. So that leads to some further questioning as far as how sites so close to each other can still have that variability that exists. So due to the subsampling occurring, we estimated the abundance as well for how many organisms were likely collected. The snags in all 3 sites had a higher estimated abundance of organisms than the riffles of the same location. Previous studies show that canopy cover does play a significant role in the relative abundance of macroinvertebrates, where streams without shading had a higher abundance than streams with shade. This is consistent with the Avery Preserve and adjacent site that have open canopy and had a higher amount of macroinvertebrates compared to Van Horn, which was characterized with about 50% canopy coverage. When we compare our EPT and non-EPT taxa, we see a greater percentage of non-EPT individuals than EPT in every site and habitat, which is an indicator for stream water quality, and we'll be looking at that in another slide in a moment. We also had a greater percentage of pollution intolerant individuals than pollution tolerant in both Avery Preserve and the adjacent site. However, there was more pollution tolerant organisms than pollution intolerant in Van Horn. There's been little evidence on canopy cover's relation to pollution intolerant and pollution tolerant macroinvertebrate colonization. Other possible factors that we can consider are the stream locations being relative to one another, where Van Horn is downstream from both Avery Preserve and the adjacent site, and the surrounding locations and fauna around each stream site. So in here, the EPT, non-EPT, PIT, and PT macroinvertebrates are compared using chi-square analysis. So they were compared to each other and to the same microhabitat of differing environments within the same location and then the varying locations. The Van Horn riffles showed significant difference between EPT and non-EPT organisms compared to the adjacent site riffle, and the pollution intolerant and pollution tolerant organisms compared to the Avery Preserve riffle. Van Horn snags had significantly different EPT and non-EPT macroinvertebrates, along with pollution intolerant and pollution tolerant macroinvertebrates from both Avery Preserve and the adjacent site locations. Macroinvertebrates have been shown to have different preferences for colonizing in different types of wood, which is a possible explanation of why the Van Horn snag was significantly different from both Avery Preserve and adjacent site snags. Overall, this is just a very good analysis of how each individual microenvironment is compared to each other and whether or not the differences that exist in EPT versus non-EPT or pollution intolerant versus pollution intolerant actually make a statistical difference rather than just an observational one. So moving on, we utilize the Illinois RiverWatch Stream Quality Ratings Index for the taxa richness, EPT taxa richness, MBI, and quality ratings determined for riffles and snags at our 3 sampled sites, as well as calculated diversity using the Shannon Diversity Index. The poor MBI quality ratings can be indicated that there has been a greater percentage of non-EPT taxa than EPT in every site and habitat. When comparing water quality to microhabitats in a single location, the quality was consistent in riffles and snags at Van Horn, but riffles showed better quality than snags at Avery Preserve and the adjacent site. This follows prior studies showing that the water quality found in riffles is usually better than other microhabitats. And then we can also conclude that taxa richness, EPT or non, is not a good indicator for water quality. As we can see, we had phenomenal taxa richness and EPT taxa richness at every microhabitat for most sites, but the water quality was not reflected of that. So for our next steps, as far as microhabitat comparison goes, the RiverWatch protocol utilizes a modified multiple habitat approach and indices to determine their stream quality. It's been recommended to develop new indices representative of individual microhabitats, specifically snags, that may better assess stream quality. For example, microinvertebrates have been shown preferences for colonizing different types of wood, and further studies might be able to determine the wood hardness of the snag sampled for macroinvertebrates to identify any possible correlations between the taxa abundance, taxa diversity, and wood hardness. Another possibility that warrants a deeper look into the protocols and indices used is the inadvertent chance of oversampling due to volunteer bias. Essentially, analyzation of the multi-habitat protocol that influenced RiverWatch found that riffles and snags were frequently oversampled by volunteers. Oversampled snags led to better water quality results, whereas oversampled riffles yielded poorer results. When comparing multiple microhabitat sampling to a single microhabitat, multi-habitats indicated poorer water quality. Many stream characteristics differ depending on their location, and creating those new protocols that acknowledge those different factors could lead to an improvement in stream quality determination with sampling macroinvertebrates. However, as everyone in the sciences know, time and funding is always fleeting. And for what we currently have now, the protocols and indices to follow, excuse me, still display a very good job of what our water quality is looking like. So now that we've covered the microhabitat comparison, I will be going over distance decay, which again was done by fellow classmate Ben Murdoch. So to begin, distance decay is essentially just decay of similarity within a community. So for our purposes specifically, we're looking at the distance decay of certain macroinvertebrates and therefore water quality to see if it's affected by environmental factors in order to help reduce possible errors in future studies. The hypothesis was that there would be very similar results at the Avery and Avery adjacent locations, while the Van Horn site would stand out from the other two, with environmental factors not playing an active role, but distance as that main determining factor. However, it was found there was little to no change between Van Horn, Avery Preserve, and the adjacent site. Environmental factors did not appear to play a major role. Distance is what did end up causing the similarities of macroinvertebrate communities. However, the environmental factors are not completely meaningless, as linear distance of a stream is being shown to be the determining factor of changes in many stream communities. And moving on to Gwen Bach's Hesterdendi research. We'll give some more background onto Hesterdendis because they're a little bit more intense and different than the prior methodology we covered. So Hesterdendis offer a passive method of sampling rather than an active method like the D-frame KIGNet used prior. It suggested that the most accurate results should include both active and passive methods to reduce the exclusion of certain macroinvertebrates. However, only using artificial substrates, such as Hesterdendis for sampling, still does provide a detailed stream community information. So this study was compared to a 2015 Hesterdendi study performed in the same location to determine if there had been a change in colonization patterns and stream quality. It is predicted that there would be improvements compared to the 2050 study due to increased numbers of EPT and PIT organisms. So 4 sets of our Hesterdendis attached to cinder blocks with clothesline and string were deployed in a V shape downstream of a boulder. Each week, one was set to be removed, sampled, and subsampled following the previous RiverWatch procedure. However, upon arrival on the second week, the remaining 3 sets had actually been removed from the water and pulled out and were up on the bank. So they were reset upstream from the boulder, and the final 2 were both removed during the 3rd week due to the setback. You'll see those denoted as 3.1 and 3.2 in the figures and images. And you can see here in our images on the right-hand side, the one on the left is our first configuration in that V shape. The second one was in more of that linear bunched pattern. So now on here we are comparing the 2021 study with the 2015 study. In the 2021 study, abundance varied week to week, whereas in 2015 the abundance drastically increased from week 1 to week 2 and continued increasing. The taxa richness and EPT taxa richness were overall less than or equal to the corresponding weeks from 2021 to 2015. The Shannon Diversity Index showed less variability in the community in the newer study, and overall quality rating was also rated less than or equal to the earlier study. Overall, it was interesting to have comparative studies performed at JJC, but external factors definitely did impact the quality of research that we did receive this past summer in 2021. And I know when I've spoken with Gwen about it, that's one thing that she would love to get back to redo to remove that environmental factor that definitely significantly impacted her work. Overall, I would really much like to thank Professor Griffiths for giving us the opportunity and guidance to be able to do this research. He really was instrumental in allowing us to be able to do all of this. I also want to thank our lab supervisors and lab tech, Christy Knoll and Kim Crow, along with JJC for all of the resources and supplies to do it. I give big thanks to Gwen and Ben, who unfortunately still couldn't be here, but for not only being phenomenal partners to work with in the field and lab, but also allowing me to present their work. And a thank you to Hannah Griffiths for organizing this presentation and allowing me the opportunity to be here and share what we did for those 6 weeks. And then if there's any questions, I'm more than happy to go through. I know we had a lot of jam-packed information there that I'd be happy to break down if anyone is looking for it.
Great. Thank you, Megan. Thank you for sharing your research and for also covering your classmates' research at the same time.
No worries.
If anybody has any questions now, you can either enter them into the chat or you can unmute yourself to ask them. It looks like in the chat, Mark is asking what a scud is. I think that's what Scud question mark means.
Yeah, so scuds were just another type of macroinvertebrate we collected. If we come back to, so the list of everything on the left is, for the most part, what we had collected and what is anticipated to collect from the RiverWatch procedure we followed. There were a few more macroinvertebrates that are in the RiverWatch procedure that we just never ran across, so they were left out because we didn't have them collected anywhere. And then we did occasionally also collect things that were outside of that list, in which there was usually a footnote that we would make at the end of each recording of just stating what it was we found that wasn't already in that list. But yeah, so scuds are these little tiny white, like probably one of the smallest ones you'll find, these little tiny white guys with a bunch of little arms, and they are everywhere. So that's what a scud is. It's just another type of one of the macroinvertebrates we were collecting.
Yeah, so scuds are actually crustaceans. Another name for them is side swimmers, so you might have heard of that before. And they basically look like little tiny shrimp.
Mm-hmm.
And then did you guys do any stream flow measurements, and did you compare it between the sites at all?
So we did take some velocities. When I was looking over Gwen's paper, because Ben and Gwen were phenomenal, they sent me all of their work and they were just like, have fun. And so with Gwen's, I know that she did note that in her report, she didn't compare the stream velocities, which she believed between the three experiments we did, hers would have been most impacted by velocity. So that was one thing where I know if she was going to pursue NCUR publication, she was going to be adding that in. But at the time, I didn't have that data in the way that she wants to analyze it for it. For my work with the microhabitat comparison, I didn't see a lot of readings that really said it impacted it one way or another. So for me, I didn't analyze that and add that in as well, because it kind of seemed like just an external factor that was there, but not very indicative of changes for me.
Okay, and then I had a question at the end of when you were talking about your own research, when you were talking about the oversampling by volunteers, what do you mean by oversampling?
Yeah, so that was actually a few articles that I came across. Mostly was one main publication that discussed it. And then it was also something that Professor Griffiths had mentioned as well that I looked into, where essentially means that when we are subsampling and taking our samples in, we're taking more than what we actually needed, and therefore skewing the numbers. So rather than it just being we're subsampling and taking that random square from our pan, and then anything that's representative that we missed, it's that we get a little carried away because we're worried that we're going to miss out on something, and then we take more than we need, which then again has shown that between riffles and snags, it can either have better water quality indications or poorer, but that's not necessarily typically what the actual result would have been.
Okay, thank you.
Yeah, no, yeah, of course. I think from the readings that I was looking into with that, it really more just looked like just more experience and training with that kind of thing, and then just having a little bit more confidence in what you are doing, so you don't feel like you have to overcompensate.
Hey, this is Jim Putz. I got a question for you.
Yeah, of course.
Why didn't you guys just sample everything that you collected at your sites and going out doing the subsampling?
Yeah, so great question. So the main reason we did it was honestly just the time constraints. Not only in general, we just had 6 weeks to be working on this, but also the amount of time that it would have taken in the field and in the lab. There were some cases where we were nearing easily close to 300 to 500 organisms based on the relative abundance calculations we did, which meant that when we pulled our samples, it's not just we pull our samples and here's all of our bugs right here in a jar. It's they're in a bucket with muck and gunk and sediment, and we have to dump them in a pan little bits at a time and slowly remove every invertebrate, and then take them back into the lab and identify. So it's just a matter of, it would have taken so much time to do hundreds of them.
Okay, because I do, I'm a RiverWatch monitor here for 20 years, and on my site, I just take everything because It's, in my opinion, I think it's a better way. You're not missing out on anything, and you don't have any what you call biases of, yeah, I might have picked the wrong square, about a random generator, and I missed out on a lot of these bugs. And they could have been over in square 1, and now they're on square 12. So
Yeah.
this is me personally. I like to take everything that and even though my site's, a pretty good site that I've been monitoring for 20 years, I take the time and effort to go through the bucket and look at everything that I can finally get out of it and put it in my jar, and then I take it to the lab and ID it as best I can. That's just my personal viewpoint.
No, I definitely agree. And I think that there are a lot of benefits to be able to doing that, especially like you said, it would remove that volunteer bias because everything that is collected are actual numbers versus us hopefully doing a good job and then having our estimated abundances. Again, I think it's just one of those things where unfortunately not everyone is able to dedicate that time for the length for collection and identification. I would love to have been able to have done that, to be honest. I would happily have sat there for hours and hours working if I was available to do it. But I definitely do agree where you're coming from, that it is a great way to remove that bias.
I got another question regarding the depth of the water in those creeks. In those pictures, it looks like it was very shallow. Were you guys sampling like inside the RiverWatch date parameters?
Yes, so that summer, here's the images you're referring to, they were actually incredibly shallow. We had a fair bit of a drought the beginning of that summer, even to the point where I think you can see right over here, that is Professor Griffiths staying there. I mean, that water is barely past his calves. And so we did take water at depth determinations. Again, I think that was more just whether it was brought into the research in this format was more based on looking into prior publications of how much it affected it. So I know for us, none of us found a lot of work supporting whether or not it made too much of a difference. So that was another one of those kind of external variables that we put on the back burner for now. But an issue that we also did have is this one mostly affected Gwen's Hester Dendy study. Again, unfortunately, she got the short end of the stick with external variables. We did catch a lot of a lot of water, which flooded the Van Horn location where her Hester Dendies were. So not only were hers pulled out after who knows how many days between that first week and then replaced, we had to pull the last 2 of them on week 3 because we had so much water come in. We just didn't know if we'd be able to make it out on week 4 to pull them. So it was at that point that water was, I don't have the depths right in front of me. I could always, if you're interested, I'm always happy to go back to my notes and send you that information. But it was pretty close to our hips and with a fairly strong current and velocity. So at that point, it was just also more of a safety concern of just we needed to get these out so we don't get more rain during the week. But we definitely did have some variations, but it was for the most part, right up until that end, very shallow water.
Okay. Because on that one picture on the left over there where you see that, you got, it looks like 3 or 4 feet of bank of the riverbed out of the water. And so looks like there's like a nice tree log laying on the ground there. It could have been a good site for stuff. Have you noticed over the course of my monitoring my site for 20 years, I noticed that in years that we have a big flood in the spring, seems to wash stuff out. Have you ever read anything or found about big flooding events? How does that determine the macro environment?
Yeah, so that's a really great question. In the research I was doing specifically, they were so focused on just comparing the microhabitats that they repeated the study so much that they tried to remove all of those environmental factors. Essentially, the more you repeat a study, the more those environmental factors become less and less of an impact. And so there was very little mention unless it was something significant of those external factors that I had found. However, we did also have at that Van Horn's location after that rain a log jam, which I did find something discussing it, stating that I believe they had a greater number of taxa collected after there was a log jam formation, simply because all the macroinvertebrates would move downstream and then kind of get stuck and then just hang out where they were at, essentially is what happened. I'm saying that one I don't remember in depth just because, again, it wasn't applicable to what I was searching for, but I know I did find something that when there was a jam or some sort where that water level was higher and there wasn't a lot of current beyond the jam, there was a significantly higher number of taxa that are collected.
Thank you.
Yeah, of course. Thank you so much for all the great questions. I'd love to pick your brain. I know you've been doing this for 20 years. I find all this so fascinating.
I wanted to jump in real quick. This is John Griffiths, and comment on some stuff with Jim, because we've done some of these studies on subsampling versus taking the total sample, and there is a little difference. What we found is that the total sample gave us a little bit more complete picture and such, and there's a lot of studies on this stuff. Some say that subsampling is as good as taking the full sample. So, a lot of variability. And then we also did something with the flooding. We had one year where we had really bad flooding with students. And what we found was that, at the time of the flood, it was difficult to sample, and it was kind of low diversity and poor quality. But as soon as the waters receded, we had a really good increase in diversity. So it appears like you're right. The flooding washes a lot of those macroinvertebrates out. But then when it goes down, you get this really good colonization afterwards. So that's what we found.
Yep. Okay, thanks.
Yeah, it's very interesting. Thank you for hopping with that, Professor Griffiths. I appreciate it.
Okay, it looks like there's another question in the chat. Have preferred water temperature ranges for macroinvertebrate species ever been determined? Would a warm stream be substantially different in diversity or EPT species than a cool or cold headwater stream?
So there definitely is work out there that I know has studied the water temperature. For us, we stayed within a pretty relative range. I want to say it was roughly about 74 to 75 degrees Fahrenheit. Again, don't quote me on that. I can always pull that at work if you guys are curious. But it was usually within a couple of degrees is what our water temperature was, also relative to the air temperature. So for us, because at least for me personally, since I was comparing two sets of samples taken at that same day and time, that water temperature didn't make too much of a difference because of how similar it was week to week. If for any reason, if we were to redo this for longer than a 6-week period and, we were going from, May to August or even pushing earlier or later in the year where there is a large increase, I would definitely, that's an avenue worth looking into, most definitely. So it's one of those, I know it's out there. There's so much information out there, and with such a short period of time, we really just had to narrow our scopes to what was most applicable to our work. So I know there is some determinations out there about whether it makes a difference. I don't have that knowledge to speak on them as of right now. But again, should we ever repeat this in the future, there'll definitely be an avenue to look into for sure.
Okay, I can add on to that a little bit. I don't know specifically like which macroinvertebrates have preferred water temperatures. However, cold water does tend to have more dissolved oxygen in it. And with more oxygen in the water, you're more likely to find those pollution intolerant species, so like the EPT species. So I can say that the temperature of the water definitely has an effect on like oxygen and other water chemistry things. So I'm sure that a lot of the organisms have a preference for different water temperatures.
Thank you, man. I appreciate that.
All right, does anybody else have any questions? Let's see here, Pat added to the chat. Oh, just that she's done some work with students comparing data throughout the years. Thank you, Pat. All right, well, if there's no other questions, I want to thank everybody for joining us tonight. Our next speaker is on February 15th. I think it's another Wednesday. It's going to be Ted Kratchmer from National Great Rivers Research and Education Center. He's going to be talking about the Lakes to Gulf program that we have at NGREC. That's basically monitoring water quality from the Great Lakes all the way down to the Gulf. And then if anybody is interested, our training workshops for the spring, most of the dates are set and they will be going up on our website on the events page and also on Facebook. And if we have any other events, you'll find them there. So thank you everybody for joining us tonight.
Yes,
And thank you. Yeah, thank you, Megan, for
I
presenting.
really appreciate the opportunity. Thank you guys.
Okay, thanks.